Basic genetics and immunohematology. So a lot of areas in a blood bank are actually going to rely on an understanding of our blood group genetics and accurate and sensitive methods of pathogen testing, and that's to help ensure to keep our blood supply safe for our patients. Most of the antigens in the blood group systems will usually follow a straightforward inheritance pattern, usually of the codominant nature. So classic genetics, as I'm sure you guys are aware, is a broad field that focuses on the understanding of the mechanisms of hereditary and variation in living organisms. It actually encompasses multiple areas, including the biochemical and biophysical nature of nucleic acids, which is the study of molecular structure and properties of DNA and RNA. Helps to understand how nucleic acids store and transmit genetic information, and it helps for insights into processes like replication, transcription, and translation, all the way down to the molecular level. Then we have population studies and epidemiology. So we have the examination of genetic variation within and between populations, study of gene frequencies, mutation rates, and how these can contribute to the population diversity, and investigation of genetic factors and disease prevalence and distribution through epidemiological approaches. And then we have understanding of inheritance patterns. This is the analysis of how traits are transmitted from parents to offspring. We have exploration of the Mendelian inheritance, which includes dominant, recessive, and sex-linked traits. We have recognition of more complex inheritance patterns, such as polygenic traits and mitochondrial inheritance. And then we also have insight on antigen typing discrepancies. So this is the identification of discrepancies in blood group antigen typing, understanding how weakened or variant alleles can alter antigen expression on red blood cells, and the application in blood cell transfusion compatibility and immunohematology to prevent adverse reactions. Population genetics. So population genetics studies the distribution and changes of allele frequencies in populations that helps to provide insights into blood group antigen variations and inheritance. There are some areas of concern, such as the work of Linnaeus and Darwin. So Carl Linnaeus developed early classification systems that laid the foundation for understanding biological diversity and grouping of species, and that indirectly influenced the concepts of human variation. Then we have Charles Darwin, who introduced the theory of natural selection and evolution, and that helps to explain how genetic traits vary over time within populations and adapt to environmental pressures. This helped to explain why certain blood group alleles may be more prevalent in specific population. Then we move on to Mendel's laws of inheritance. So Gregor Mendel established the basic principles of inheritance, including the concepts of dominant and recessive traits, segregation, and independent assortment. These laws are fundamental for understanding how blood group antigens are actually inherited and can help predict blood type inheritance patterns within families. Hardy-Weinberg principle, so it is actually a mathematical model that describes the genetic equilibrium within a large randomly mating population where allele frequencies remain constant over generations in the absence of any evolutionary forces. It helped to provide a baseline to detect when allele frequencies are changing due to selection, mutation, migration, or a genetic drift. This is useful in blood banking to estimate genotype and phenotype frequencies for blood groups and populations and predict compatibility risk. And we also have inheritance patterns. So this is the study of how different blood group alleles are transmitted through generations, including autosomal dominant, autosomal recessive, codominant, and sex-linked inheritance patterns. Also, understanding these patterns can help aid in resolving blood typing discrepancies and managing transfusions, especially in complex cases that can involve weak or variant alleles. So Mendel's first law in blood group genetics, his first law was law of segregation. This law states that alleles of a gene pair separate independently during the formation of the gametes, so the eggs and the sperm. So each gamete carries one allele for each gene. So importantly, the alleles themselves do not alter each other when they coexist in the same organism. They segregate unchanged into different gametes. So application from this to the blood group genes. So unlike traits such as flower color in many plants where one allele may be dominant over another, most blood group genes exhibit codominant inheritance. So codominance means that both alleles in a heterozygous individual are fully expressed. So for example, in the ABO blood group system, individuals inheriting both A and B alleles will express both antigens on their red blood cells, which will result in an AB blood type. Some implications, so understanding codominance in blood group inheritance is crucial for accurate blood typing, transfusion compatibility, and predicting inheritance patterns in families. Codominance, as mentioned before, both alleles are fully expressed, and the products of each allele are visible at the phenotypic level. This means that neither allele masks the other expression of the other. So an example of this is we have the MNS blood group system, which we'll talk about later on in the course. But individuals who are heterozygous for the M and N alleles, so they have a genotype of MN, will express both the M and the N antigen on their red blood cells. So these individuals will type as positive for both M and N antigens, and it will demonstrate the codominant nature of these alleles. The significance of this is understanding the codominance and that it's important for blood typing, transfusion compatibility, and predicting antigen presence in your donors and recipients. Second law, Mendel's second law, is the law of independent assortment. Mendel's second law states that genes for different traits are inherited independently from one another. This means the inheritance of one trait does not influence the inheritance of another. So the implication of this, so this law allows for all possible combinations of alleles for different traits to occur in the offspring, which helps to increase genetic diversity. So applicability for this in the blood bank, Mendel's law applies to all sexually reproducing diploid organisms, including humans, which makes these principles relevant in genetics and blood banking. Gene linkage and its effect on inheritance. So gene linkage, these genes are located close together on the same chromosome and tend to be inherited together as a single unit, and this is known as a linkage. So the effect on Mendelian ratios, when genes are linked, the classic Mendelian ratios expected from independent assortment in the F1 progeny may not be observed. This is because linked genes do not assort independently during meiosis. So there are, of course, some exceptions and considerations. So sometimes even linked genes can be separated by recombination or crossing over during meiosis, which can shuffle gene combinations. So the closer the genes are on the chromosome, the less likely recombination will occur between them. Linkage can cause deviations from expected inheritance patterns and complicate genetic predictions. So the importance in blood group genetics for this, so linkage is especially important in blood group genetics where certain antigen genes may be closely linked, which can actually influence inheritance and phenotypic expression. All right, so the effect of recombination. Differences in the gene ratios of progeny from F1 matings can occur in recombination if recombination takes place during meiosis. So recombination is the process, once again, where homologous chromosomes exchange segments, potentially separating linked genes. An example of this in blood bank would be, again, the MNS system. The M and N alleles and the S alleles are located close to each other on the same chromosome and tend to be inherited together due to linkage. However, because recombination can occur during meiosis, these alleles may sometimes be separated, which can lead to a different gene combination than expected if these genes assorted completely independently. So understanding the linkage and recombination is important in blood banking for accurate prediction of inheritance patterns, especially for linkage blood group systems like the MNS. Hardy-Weinberg principle is a fundamental concept in population genetics that allows for the detailed study of Mendelian inheritance within large populations. This principle provides a mathematical model to calculate and predict the genetic variation of a population under ideal conditions, assuming no evolutionary influences such as mutation, migration, selection, or genetic drift occur. The Hardy-Weinberg formula is expressed as P plus Q in parentheses, and then another set of parentheses, P plus Q equals 1. The P equals the gene frequency of the dominant allele, and the Q is the frequency of the recessive allele. So this equation allows for prediction of genotype frequencies based on allele frequencies and is essential for understanding how traits are passed down in populations. The principle specifically addresses questions about recessive traits and how these traits can persist in population over time, even when a recessive trait is not visibly expressed in individual, so like it's hidden in heterozygotes, it can remain present in the gene pool at stable frequencies. This helps explain why certain recessive genetic disorders or traits can remain prevalent within populations despite not being commonly expressed phenotypically. So the criteria to be met for the equations to be using these appropriately, there are certain conditions that must be met for the Hardy-Weinberg equations to be valid and used appropriately. So the first one is the population studied must be large, and that helps to minimize random genetic drift. Mating must be random, so no mate preference based on genotype. No mutations occur in parents or offspring, and no migration in or out of the population. No differential fertility or mortality of genotypes. So all genotypes have equal chances of survival and reproduction. Some problems with the Hardy-Weinberg criteria. So collecting sample data from a significantly large enough segment of a population is just not always feasible. Mating is not always random in natural populations. And mixing of populations on a global scale can lead to gene flow on a constant basis, which can affect allele frequencies across the populations. So some pedigree analysis. So males are represented as squares, females are represented as circles, and a horizontal line joining a male and female will indicate a mating between the two for these pedigree analysis. Some additional pedigree conventions is offspring are indicated by a vertical line descending from the mating line. A double line between a male and a female indicates a mating between relatives. A small black circle represents a stillbirth or abortion, and deceased family members have a line crossed through them. The proboscis is called the proband, is the most interesting or important member of the pedigree. It is indicated by an arrow pointing to that individual. Often, something unusual about them is the reason the pedigree analysis is undertaken. So they're kind of like, I don't know, patient zero, I guess, if you want to go that route. So this is an example of what a pedigree would look like. Cellular genetics, so humans have 46 chromosomes, 22 autosomes, and one set of sex chromosomes. The chromatin is a nucleic acid, and structural proteins are called histones. The specific location of a gene on a chromosome is called a locus, or a loci for plural. And at each locus, there may only be one or several different forms of the gene, which are going to be called alleles. Phenotype and genotype. So the phenotype is the observable characteristic or trait of an organism, where the genotype is the genetic makeup of the organism, or the combination of the alleles. We have homozygous and heterozygous. So homozygous means that they have two identical alleles for a particular gene, and heterozygous means that they have two different alleles for a particular gene. There's also a gene called the silent gene, or an amorph, and this is a gene that produces no functional product, so it just kind of is there. Chromosomes are composed of long linear strands of DNA, and the DNA is tightly coiled around basic proteins called histones, as shown in the picture below. DNA is made up of two types of nitrogenous bases. First one is purines, which is made up of adenine and guanine, and then you have pyrimidines, which are made up of cytosine and thymine. So DNA is a double helix, as I'm sure you are aware, and it's composed of two strands coiled around each other. The strands are complementary and anti-parallel, meaning they run in opposite directions. It has a sugar phosphate backbone that forms the outside of the helix, and it has nitrogenous bases form the interior rungs that pair specifically with each other. So the adenine and thymine go together, and then the guanine and cytosine go together. There are certain common approaches in modern genetic techniques. So you can do amplification, visualization, isolation of material, things like that. And the role of genetics for the future, so this is always an evolving field. It has DNA-based application for transfusion medicine, For genetic variation, it can impact response to many types of treatment and also personalized medicine. So, treatment and prevention of certain diseases for that person based on their genes, environment, and lifestyle.